Thank you. Thank you for having me at the SSG. Wonderful meeting. And this is just a little bit of the work that we've done to set up some tools for research, for translational research in the laboratory. And so why do we need to preserve the tissue? Why is it important to have good quality tissue? As there's three reasons here. The molecular diagnosis of tumors has been evolving to be really what dictates the diagnosis. In the future, personalized medicine is going to become important, and characterizing the tissue well, in whether it's biological behavior or genetic abnormalities, is very important. And also the research that all of us are very interested in doing for our patients. And so just as background, glioblastoma, obviously everyone knows that it's the most malignant tumor, most malignant brain tumor with poor survival. Other tumors that we are working on include all these ones that are listed there. And so what is important of preserving the tissue in the new era of molecular diagnosis? And this is what we always deal with. The histopathological diagnosis is subjective, although neuropathologists can become very experienced and give us very accurate diagnoses. It's subjective. We have a very heterogeneous population of brain tumors, even within the GBM population of brain tumors. And we also want to know the characteristics, cellular characteristics of individual cells. Now, molecular markers nowadays can classify GBMs into all these different categories: IDH mutant, IDH wild type, the proneural, the neural, the classical, and the mesenchymal. And this also has treatment implications, where tumors that have X or Y diagnosis have to be funneled to different treatment modalities to help with better outcomes and better quality of life, better progression-free survival, and so on. Meningiomas, on the other side, are mostly benign tumors, with a small percentage of them being aggressive grade 2s and grade 3s that have higher grades of recurrence. They also have a lot of molecular differences depending on the anatomical location. The grade also portends prognostic indications with the molecular diagnosis. And so preserving tissue in these tumors and all the other ones that I mentioned is very important, because it can guide us in terms of the treatment, in terms of the prognosis for the patients, in terms of the outcome that we're gonna be able to achieve with a patient, and so on. And so what is very important is that we have very good quality tissue that represents the actual characteristics of the parent tumor while it's in the patient's brain. And how do we do that? We use to resect these tumors, we use mostly these two devices, the Nico Myriad or microdebrider. We also use the Sonopet or the CUSA, which are ultrasonic aspirators. And so not everyone uses the Nico, not everyone uses the Sonopet. So how do we know if the tissue that we're going to acquire from any of these devices is good for research? So we had this hypothesis that the Nico, because it's a microdebrider, it gives us how do you say this? Unmodified tissue that has live cells and gives us good quality tissue, because it just suctions the tissue into the aperture of the debrider and then cuts it into small pieces, thereby preserving cells in the middle. Whereas the Sonopet or any ultrasonic aspirator, in theory, sonicates the tissue, it breaks it down, and then it sucks it up. And so when we collect the tissue from the automated collection system, from the Nico Myriad, we get these nice little pieces of tumor that are easy to deal with. Whereas when we collect the tissue from the collection system that we use for the Sonopet, which is like a sock that we put into the suction cup, it looks like a slime more than any formed tissue. And so the hypothesis was the Nico Myriad will give us better tissue for molecular diagnosis, better genetic material, better cells to make better cell lines, better organoids, and so on. And so we put this to the test. We collected the specimens from the same patient using the two different modalities of resection. only the patients that required the two for the resection, whether the tumor was too hard for one or the other. Um, we left this to the discretion of the surgeon collecting the tissue. And then we processed the tissue the same on both devices. And we measured cell viability. We sent the samples to pathology, whether it was the Sonopet or the NICO. We did cell culture to generate cell lines, neurosphere cell lines and adherent cell lines from these tumors. We isolated RNA and tested the quality of the RNA to determine whether this was degraded or not. And so through all this, we're also building a live biobank of brain tumors for research in terms of tumor biology. And at the same time, we're getting immune cells from the tumors and from the patient's blood to build a comprehensive live biobank for future research. So this is our workflow. We get the tumor, we process the tissue to make cultured cell lines. On the other hand, we also process the tissue and the blood to obtain the infiltrating lymphocytes and the peripheral blood, the PBMCs, the peripheral blood monocytes, and we save everything. But in the downstream assays that we devised for this project is that we wanted to know what the tissue looked like in the cell line, in a histopathological analysis, and molecularly using RNA extraction. And so first hypothesis is that the cellular viability of the cells that we obtain from the Sonopet is going to be lower than the NICO because it's obvious that that's the mechanism is going to happen. Surprisingly, after measuring mean viability from both samples, even though there is some variability between samples, there was no difference overall in all the tumors that we tested. And when we tested the tumors for each specific histology, GBMs, meningiomas, pituitary adenomas, chordomas, and metastases, there was no difference either. Histopathologically, we sent samples from the Myriad and from the Sonopet to have a good H&E comparison of the pathological anatomy of these tumors. And we discussed it with our pathology partners, and same, there was no difference, surprisingly, in the appearance of the tissue. RNA concentration, how much RNA can we get from both samples? Comparison showed no difference. The PCR analysis of a few housekeeping genes to determine if the quality of the RNA was optimal for further molecular research. And so we tested these 4 housekeeping genes, and also we did not find any significant difference or trend in the quality of the RNA that we obtained. This is by histology. Same thing, no difference, no difference except in the chordomas that we had 3 tumors only. We were not able to do statistical analysis in that cohort of patients. And so we decided to then move on to the next project. And so how can we apply these technologies to get better translational research systems? And we looked at the organoids as a means of maintaining cellular heterogeneity for GBMs and other tumors. And so how do we make the process of culturing these tumors as organoids more efficient, more efficacious, and to make the workflow better? And so we used we want to use the organoid system because it recapitulates the heterogeneity of the tumor. It has its own microenvironment, even though some of the immune cells start fading away by 2-3 weeks of culture. And so we started with the process that we were using before, where we get the chunk of the tumor, we manually divide that tumor into small pieces, about 2 to 3 millimeters. And then we put that in culture, and then we get these awesome organoids that look like spheres, but they actually have a very good amount of cellular heterogeneity and architectural heterogeneity that resemble the tumor. One of our residents trained in his PhD at Hopkins, and he was in the lab that started pioneering all these organoids, and so he's been very excited to work with us creating these systems. when we discussed the project to use the microdebrider, the nicomyriad, to get the little chunks of tissue and avoid all the mincing and dissecting of the tumors, he was very excited and immediately jumped into the idea of using this modality for the generation of the GBOs. And so we wanted to see if it was feasible, if the tissue that we obtained was enough or as good as the manual dissection. And so up to now, we have 6 paired specimens from GBM and meningioma patients. That's another thing. I'll show you in a little bit. Using the nicomyriad to grow organoids with meningioma patients, surprisingly, meningioma organoids grow, which is in and of itself pretty amazing because these tumors don't grow in cell culture. And so that gives us another option to do translational research in primary human tumors from meningioma patients. But let's go to see what we have optimized in terms of this workflow. The processing time is significantly less when we use the myriad versus when we have to dissect tissue by ourselves. For obvious reasons, we already get the minced tissue in meningiomas and GBMs. The cell viability is equivalent, and I would say maybe a little bit better because these organoids start growing a little bit faster, start rounding up a little bit faster, which is a measure of their health. And so we did these organoids and compared their growth at 4 weeks and at 3 months. They grow pretty similarly, but like I was saying, even though it's hard to quantify this, the myriad derived organoids round up faster and we have to divide them faster. And so how do they look, these organoids under the H&E stains? They show very similar histopathological diagnosis to the parent tissue. And you can see the GBMs have the cellular heterogeneity as compared to the en bloc tissue. The meningiomas looks the same. Meningiomas are sometimes more fibrous, and these fibers also get perpetuated in the organoids. Another image of these organoids at 4 weeks compared to the parent tumor. And something that is very important and key when using these tissue systems for research is, do the cells proliferate? Can you recapitulate the amount of stem cells that you have in these organoids compared to the tumor? And how do they look in general? And so these are just some immunofluorescence images showing Ki67 labeling of these tumors, showing that the tumors proliferate or the cells proliferate while in culture. They also show SOX2 and nestin markers for stem cells. We show the characteristic GFAP staining that all astrocytic tumors have. And obviously the montage of the 3 images, and you can see how the periphery of the organoid is more active in terms of division, cellular division, than the inside of these organoids. And that may be related to the hypoxia that the center of the organoid is experiencing. And that brings other questions, other things that we can tackle using this organoid system. And in the terms of meningioma, you can see, I hope this is projecting as good as it is on the screen, but these meningioma organoids stain for the progesterone receptor that is commonly found in meningiomas. We also have Ki67 labeling of the cells. That means they're dividing in vitro. And you can see that there's many other cells that are derived from these meningioma organoids that don't stay for these other markers, but they're important to know that they're there because they can be fibroblasts or other tumor-associated cells that are active in the microenvironment. And so this is just how we established a tool in the laboratory to be able to further study these tumors. For example, in the GBM using the organoids, we're going to start looking at stem cell factors that are canonical in other cancers that we can knock down or overexpress to see if the stem cell compartment of the tumors can be quenched. Personalized medicine, as the company Kiatech has been doing, screening these tumors for different types of chemotherapeutic agents with their assays, testing them in organoids that better recapitulate the tumor as opposed to the cell lines. Molecular diagnosis is extremely important. And for me, a personal, very big interest is the research. That's all I got. I want to thank the SSG organization for inviting me and all these people on this slide that make up the Emory Skull-Base Center. I'd be happy to take any questions.
I'm just curious with the ultrasonic aspirator. I'm looking at those tissue specimens. I'm actually surprised that they maintain this cellular architecture. Were you using this primarily on suction, or how much energy were you having to use when aspirating?
That's a really good question. Everyone that has used an ultrasonic aspirator on a fibrous meningioma knows that if you put it to 100%, you can even see it burns the tissue. So for these tumors, the consistency of the tumors is very important. And we always used 50% power on the sonoping. We didn't go above that because we know that we can create thermal injury. And I believe some people have done measurements of what the temperature at the tip is, and depending on the irrigation. So there's a lot of factors to control that. That's why we kept it 50, 50, 15, which is what we use for all these cases that we collect. All right. Thank you very much.